All right, off we go.
So just as was previously said, I believe we can all relate to the struggle of data volumes increasing, and this has made it harder to visualize big data as there's no standard way of doing things with various file formats and viewers available. Because I focus on accessibility, I compare active, free and open source viewers based on features which I believe are integral to the visualization of big data. These features include multi-dimensional support, orthogonal viewing planes to interrogate within the volume, the ability to overlay segmentation labels, and next-generation file format support. I go into more detail about how each of these viewers compare in my poster, but long story short, Nopari seems to be a suitable approach for now. The question is now how to put this into practice. Is it easy to use and implement with big data, and can we package it up with a nice workflow for an end user? Alongside Nopari is a suggestion of using Dask to speed up the bottleneck of reading and writing the data. The multi-dimensional data is chunked and streamed into RAM as and when required by the viewer to display data such as a 6-channel 30GB file. Dask is fairly easy to integrate with Nopari, but we want to be able to optimize the reading and writing to maximize interactivity so that the user can freely rotate a volume and navigate the time series with minimal delay or lag, which would be exaggerated using a remote or cloud-based system. This will depend on features such as the number of chunks, the shape and size of chunks, and the operating system you use. I explore some of these parameters, and you can find my benchmarking code as well as my poster at this GitHub repo. We've all had to view data at some point, so I'm sure have opinions about the most ideal approach to the problem and what you want from a big data viewer. So please come to poster 6 to share your thoughts.
Alessandro.
Hi everyone, I'm Alessandro. I'm the software developer at the Sainsbury Welcome Center. I work on the BrainGlobe tools, which are a set of open source, a collection of open source tools written in Python. We have lots of Nopari plugins as well for computational neuroanatomy. We try to analyze data in kind of a common coordinate system, and mostly my poster is about how we make life easier both for users of our software, but also how to make it easier to contribute to our software. And I'm not going to spoil anything, but come see me at poster 8 to see how we balance that trade-off between using and developing software.
Okay, Elaine.
Cool. Hi everyone, I'm Elaine. I'm a research software engineer at the Rosalind Franklin Institute. My poster is on building reusable bioimage analysis tools for other people. So for context, I work in a team of research software engineers, and we build things for the cryo-electron tomography community that's built up of structural biologists, biochemists, microscopists, lots of other people. And we built Autorac, which is a pipeline tool to join the different separate software packages that don't talk to each other and generate reports to look at the results. But through this kind of process, we discovered a lot of issues that many people would relate to, like how do I install this on my computer? How do we train users to use our data? And how do we maintain and update our software as we go along? So we kind of wanted to distill the lessons that we learned through developing this first version of our software, which are that user engagement and good communication was really important throughout the development, and good software engineering was definitely worth the effort. But because we're here at CBIAS and everyone's here from different fields, we'd like to hear from you in my interactive poster at poster 14, where I'm going to be collecting ideas of all the discussions that we're going to have. And yeah, hopefully kind of share it later so we can all build better reusable tools for better bioimage analysis.
Great, thank you.
Hello everyone, I'm Joachim Lindblad. I'm a professor in computerized image processing at the Center for Image Analysis in Uppsala. And I'm an algorithm developer. So this morning we all heard how useful it is to work with multiple modalities to do correlative imaging and fuse the different modalities in order to learn more about an imaging sample. And in order to do that, we typically need some kind of multimodal alignment to put our images in a common framework. So what I'm presenting at poster 19 is a clever trick that one of our PhD students figured out, how to compute mutual information in the Fourier domain, such that we can actually compute it similar to cross correlation, but this as a cross mutual information. So the mutual information for two images translated all possible translations very quickly in the Fourier domain. And since it's so fast, this allows us to do kind of grid search approaches to find very nice registration, multimodal registration performance. And it's also a very nice and elegant algorithm. And I think it's, everyone can implement it. And just made a small implementation in MATLAB, which is very easy to follow. In addition to that, I'm also squeezing into my poster one application example where we are using this for inside a browser, which is a web platform that we have developed for collaborative annotation and viewing of whole slide images, so 100,000 times 100,000 pixels. So please come and see me at poster 19. Thank you.
So, Joel.
Hi everyone, my name's Joel, or that Zurich guy. You can find me at poster 20 to come chat with me. My poster is all about the fractal framework that we've built. It's a framework that is useful to you if you want to process OMSR images. It specifically shines if you either have big 3D images or terabytes of fluorescent images. We built a modular system for workflow building and controlling it from a web interface. And you can learn more at the poster. The thing I can show you at the poster are movies. So here are two movies. On the right, you see our web interface that makes it easy for people to add tasks to a workflow, build up what they want to process, change the parameters, and submit it to a Slurm cluster. And on the right, you see us browsing big OMSR files actually in Napari. So I'm curious about poster 6 and the benefits you have of using this new file format, browsing large data amounts. Come by at poster 20 if you want to learn more.
Noriko.
I'm Haruhiko, undergraduate student from Nagoya University. Two photomicroscopy image browse in axial direction, making it difficult to analyze three-dimensional structures. Commonly, deconvolution methods are used to develop these images. However, these methods require extensive hyperparameter tuning, bandwidth noise, and often fall short in achieving sufficient resolution. In this study, we developed a deep neural network that develops and improves the resolution of DPM images in an unsupervised manner. Initially, we trained the developing model to solve the inverse problem of DPM image acquisition process. This process was approximated by a physics-based generated model. Subsequently, we optimized both the developing model and the physics-based model, aiming to minimize the loss between the real input images and their reconstructed counterparts. Our model was tested on actual images of beads, and we found that our model significantly improved the accuracy of volume estimation. We also adapted our model to TPM images of mouse brains, and we found that our model successfully inferred precise three-dimensional structures. We hope that our model will aid in observing microscopic structures which have been challenging for conventional methods. If you are interested in my method, please visit us at poster number 24. Thank you for listening.
Hello, everyone. I feel like a sore thumb because everybody's developing really cool tools, and I will show you some biology and how to use image analysis. So my project is about how to use image analysis to guide tissue proteomics in triple-negative breast cancer, one of the worst subtypes of breast cancer, because we do not have good enough targeted therapies, And when patients get treated with chemotherapy, resistance arises very early. The cohort I'll be working on is about 88 patients, and we have samples that have been collected in FFPE before and after a chemotherapy. These samples have been, or then the process of being imaged with multiplex imaging in tissue microarrays with 40 markers at 40x in a confocal system. And this is to distinguish all the important cell types in the tumor microenvironment: immune, cancer, and even and fibroblasts. I'll be using, as you have heard from today, MC Micro, which is a Nextflow pipeline to pre-process, stitch, segment, and analyze all of these data sets. And after I've performed the analysis, I will look into what are the most interesting interactions in these tissues between the respondents and non-respondents of treatment. And then we will collect those cell types from those tissues with laser microdissection. Those cells will be pooled, and then they will be processed and run with the best mass spectrometry money can buy. We plan to collect from 50 to 100 cells for each cell type, and then get from 4,000 to 5,000 proteins in a readout. Hopefully, understanding what are the differences between the cell types and those cell types between the different patient treatment outcomes, we can better understand resistance and provide better clinical decision-making for clinicians and better treatment options for our patients. Thank you very much, and I'll answer any questions at poster 26. Thank you.
Cameron.
Hi, I'm Cameron Shand. I'm in the software engineering and AI team here at the CRIC. And we've been developing something called AI on Demand, which ultimately is about empowering scientists and analysts to use the latest machine learning, deep learning models using HPC as part of their usual workflow. So at a very high level, we've developed an Apari plugin that goes into a Nextflow pipeline, which fortunately Christian introduced. And out of it, you get your results. So the main aims of the projects are to effortlessly run pre-trained segmentation models through an Apari plugin, to easily incorporate new or user-contributed models so that then the community can kind of contribute to this so your models can be available to other people if you want. Also to enable fine-tuning for task-specific models, because a lot of the models will not work on your data out of the box, and to enable compute-agnostic execution, which we get for free thanks to Nextflow. Progress is reasonable so far. It's currently being tested internally here at the CRIC, so it's not just me using it. A bit of progress on user-contributed models, but I think the thing I really want to highlight is this sort of fine-tuning. It's currently in development, but I think it's really important for sort of vision foundation models like Segment Anything. So how does it look? So this is the simple Apari plugin on the left-hand side. So you select the organelle, essentially the particular task that you want to segment. That then determines which models are available to you. You can modify the parameters if you want. Select your data and then execute the pipeline, which currently we're hosting it on demand. So then it just plugs into the CRIC HPC, but thanks to Nextflow, it can work on a lot of things. Each mask that gets segmented slice by slice is fed back live into Apari, so you can view the results as they come in. Currently, we have a couple of internally based, UNet-based models, internally developed, sorry, as well as MitoNet and Segment Anything, but hopefully we'll expand more models in the future. Thank you.
Daniel.
Hello, I'm Dan Stedman, so I'm a PhD student here at the CRIC. I spend most of my time doing experimental work with a couple of techniques that are super resolution microscopy techniques, so DStorm and DNA Paint. And these techniques are really powerful to study the nanoscale architecture of protein complexes. But one of the problems that you run into is that you often have incomplete labeling. So you have missing sites in your protein architecture on just one copy of your protein complex, and you also have issues with localization scatter because there's some imprecision in your experiment. So what I'm aiming to do with what I'm presenting at poster number 40 is to aggregate information from many copies of the same protein complex to build up a more accurate model of that protein complex. So you could take many images of nuclear pore complexes and then aggregate them together to get out a more accurate representation of the nuclear pore complex structure. So to do this, I use a convolutional neural network, which estimates the pose of input images relative to a point cloud model, which is the thing represented in pink. And then you can train this system, and what you're doing here is you're optimizing the point cloud model so that it ends up representing your biological structure. And to get 3D information in, I input 3 renderings from 3 different faces of a cube. And just some kind of preliminary results. You can see some more if you come to the poster at poster number 40. You can pull out nuclear pore complexes with 8 sites with the 8-fold symmetry. Video might play. Yeah. And see 2 rings. And then if you look at a single ring, you can pull out 8-fold symmetry and look at the spacing between rings. So come to poster number 40.
Thank you very much. Last but certainly not least.
Hi, I'm Gebhard. I'm a computer scientist embedded in a biology lab. And we're interested in tumor immunology. So we investigate how killer cells kill cancer cells. And to do that, we stain our cells, the cancer cells, with a FRET sensor, so we can analyze the cell death, and the killer cells with a cell tracker deep red. My part is developing a interactive, biologist-friendly application that allows them to analyze all the cells, to do cell tracking, background correction, and of course, fluorescence analysis to get a classification for the type of cell death. And my poster is number 42, as Marvin tells you. This is my team. We're having quite some fun at conferences. Please come talk to me at poster 42. Thanks.